Antigens and antibodies. Let's take a look at the exterior of the cell. The outside layer of a cell is composed of a lipid bilayer membrane. There's two layers of fats, and they're arranged so that you have a hydrophilic, that's a water-loving side facing out, the outside of the cell. Cells are always in an aqueous type of a solution. Your body is wet, the cultures are wet, animals are wet. So everything that you see in life is going to be in water in some way. So the water-loving parts of the lipid bilayer membrane are facing the outside. You get the picture. And then the water-fearing side of this molecule is facing the inside. The fatty side, the water-fearing side, the side that repels water, faces each other. They're passively attracted to one another. In this lipid bilayer, which sounds kind of flimsy, like you have a bunch of this stuff just sort of stuck to each other without any kind of extra glue or staples or anything like that, it's actually quite secure. Embedded in this, you have all kinds of other molecules. You might have channels that can allow ions or sugars or hormones to pass in and out. And on the outside, you can have any variety of different sugars or proteins with sugars on them, or fatty substances with proteins and sugars on them. There's all kinds of different markers that you can have on these cells. These tags are on the outside. And they have functions of their own. But one of the important things that we care about is that the combination of stuff on the outside of these cells are genetically driven. Your genes dictate what these outside of these cells look like. And what the outside of these cells look like is something that your immune system can recognize, so that your immune system knows what cells are yours and what cells are not yours, and therefore what cells should be attacked as foreign invaders. All cells have these tags. These tags are called antigens. Again, antigens mark cells as being either self or non-self for your immune system. When your immune system is chugging along and comes, a white blood cell, a leukocyte, is chugging along and comes across an erythrocyte, a red blood cell, it can sort of do a quick scan of the outside of the red blood cell and say, oh, you're Rhiannon Robinson's red blood cell. I'll leave you alone and move on. And if it chugs along and comes across something else, like a bacterium or your red blood cell, it'll go, oh no, I can't tolerate that. I'm going to have to attack it and get rid of it. It's not part of the self. It has to go. Foreign cells are attacked by the immune system in this way. Now, the actual mechanisms that your body goes through to counteract any kind of foreign invader in your body is super, super, super complicated. It's whole, degrees unto itself, let alone a portion of a class. What we're going to suffice to say is one of these components are called antibodies. Antibodies are attracted very, very, very specifically to specific antigens. So you can see the ends of an antibody here has a given shape, right? This shape is only going to fit the type of antigen that'll fit in it. It can't fit the other things. So one type of antibody will only attack at one type of antigen. Now, what really sort of happens with this, it's not so much an attack. The antibody will latch on to the antigen, that'll activate it, and then it'll travel around and stick itself to the outside of a bigger white blood cell so that the white blood cell can recognize these foreign invaders and then make even more antibodies against that specific thing. They clump up, they attach to other cells and neutralize them. In forensics, we're mostly concerned about the antigens on the outside of erythrocytes, that's white blood cells. All erythrocytes have self-antigens, that's the antigens that say that I am part of the same body, plus a couple others, whose role we're not totally sure of, but we know how we can exploit them anyway. erythrocytes have all the regular antigens plus A antigen, B antigen, and may or may not have Rh factor. When you see that somebody's blood type is something something positive or something something negative, the plus and minus indicates the presence or absence of Rh factor. So if your blood type is O negative, for example, you do not have Rh factor on your blood. If you're O positive, you do have Rh factor in your blood. The type of these these antigens are inherited from your parents. So your blood type is determined by what your parents' blood type is. And it's highly figure-outable as to whether or not, or what possible blood types a person could have if they were to have children with somebody else. I'll show you how to do that later. Blood type antigens are co-dominant alleles. We're going to talk about different modes of inheritance later on, but suffice it to say, what your mom gives you and what your dad gives you, mom's blood type and dad's blood type gene, both show up in your blood. Again, that's a super, super loose way of describing it, but for our purposes for now, until we explain it later on, this will suffice. You get genes for your blood type from your mom and a gene for your blood type from your dad, and together they both show up and make your blood type. So for example, if you got an A antigen from mom and a B antigen from dad, your blood type would be AB. If you got an A antigen from mom and a B antigen from dad, you would have, and another A antigen from dad, you would have type A blood. If either one of them had a positive blood type, then you would also, and they gave that to you, then you would also be something positive. If neither of them had Rh factor on their blood, then you would be definitely negative. The absence of A antigen or B antigen is type O. You can think of an O like 0, like there are 0 antigens on somebody's blood, but it is actually the letter O, like orange. So you may have an A antigen, you may have a B antigen, you may or may not have Rh factor, you might not have any of these things. If you have no A antigen, no B antigen, and no Rh factor, your blood type is O negative. If you have, let's do one and I'm going to pause for you to help figure it out. If you have an A antigen, your mom gives you an A antigen, and your dad gives you a B antigen, and your dad also has Rh factor, what's your blood type? AB positive. Mom has A, dad has B, dad also has Rh. I'll give you a little bit of a trickier one now. What if your mom has A, your dad has O, but your dad also has Rh factor? What do you think it would be? Mom is A, dad has no antigen, but he does have the Rh factor. If you guessed A positive, you're correct. You'll notice there's no AO blood type listed here. When you don't have something, blood type O is no antigen. When you don't have something, you don't have anything that can hide what the other parent has given you. So if one parent has a lettered antigen and the other one has no antigen, the lettered antigen is what's going to show up. The ABO system is separate from Rh factor. They're different genes. So you can have blood type O and also have Rh factor. That would be O positive. This is very, 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 very easily characterized in lab. These blood types, these antigens, are useful to us because they can provoke a very, very strong immune response when injected into another person's blood. More casually phrased, if you inject somebody's a different blood type into somebody's bloodstream, their immune system is going to freak out. They're going to see that blood coming in and they're going to go into big time, clot it up and attack it mode. It provokes a very, very strong immune response. So what you can do is you can effectively reproduce that same response on a very small scale in lab using evidence blood and antibodies that you have manufactured for you, expose them to one another, and you can see what a person's blood type is. All the blood types that are possible are listed right here. So your blood type determines what antibodies you do have. Remember, you have antibodies that scout your bloodstream and look for foreign invaders. You also have blood cells bopping around in your bloodstream with their own antigens. If I am a red blood cell with the B type antigen on me, do you think it would be useful for me to have anti-B antibodies? In other words, if I have type B blood, would it be good for me to have antibodies that will attack type B? No, that would be terrible. I would clot to death immediately and I would just die without any chance. All of my blood would go bad. So, or would be attacked by my own antibodies. So that wouldn't be good. What my bloodstream does have is antibodies that attack the other antigen. So I personally, and this is true, I have type B negative blood. So my erythrocytes are bopping around with these B antigens, these aren't the real shapes, attached to the outside of the cell. You can see how its shape is different from group A. In my bloodstream, I have antibodies that attack type A blood. My antigens are B. My antibodies are anti-A. This can be very confusing. They both sound very, very, very similar. So I'm just going to restate it one more time. Antigen is my cell tag. Antibodies equals immune attackers. Okay? Group B blood has anti-A antibodies. Group A blood has anti-B antibodies. Group AB blood has both A antigens on it and B antigens on it. If it had anti-A antibodies, you'd be attacking yourself. If you had anti-B antibodies, you would also be attacking yourself. So group AB blood has no antibodies that are going to attack A's or B's. Group O, if you recall, has no antigens. You see how there's nothing at the end of these spikes? It's just a naked cell. So it has no antigens on it. So anything that comes in with an antigen is foreign. Therefore, you make both types of antibodies. Again, with Rh factor, that's its separate thing. If you have Rh factor, you will also have Rh on the outside of your cells, and you will have no anti-Rh antibodies. If you are Rh negative, you just don't have anything on the outside, then you will have anti-Rh antibodies. Practically speaking, this matters when it comes to transfusions and blood donation. What blood type you have will determine what types of blood you can receive in a donation and who can get your donated blood. This chart boils it down quite nicely. If you have, for example, like let's just stick with me here for a minute. I have type B negative blood. I can donate to anybody who does not have anti-B antibodies. Type AB, they have a B in their name, so they're not going to have antibodies to attack Bs, so I can donate to them. I have no RH factor, so there's no RH for anyone to attack, so I can donate to pluses or minuses, positives or negatives. I can donate to AB negative as well. I can donate to other B negatives. I can donate to B positive. Now think for a minute. Why can't I donate to type A negative blood? We're both negative. Right. It's because type A blood will have anti-B antibodies. If they get my blood, they'll attack my blood and they'll be just as sick as they were before. What about O positive blood? They're not going to have any RH factor antibodies. So my negative doesn't really matter one way or the other. But they'll also have anti-B antibodies, won't they? So they can't receive my blood. Now, who could I, as a B negative person, receive blood from? I can receive any blood that has the B antigen and no RH factor. If I get somebody with positive type blood, they have RH factor, my bloodstream is going to have anti-RH antibodies. It'll go attack, attack, attack. If you give me A's, my body will go attack, attack, attack. It can only take what it already has or just can't recognize because there's nothing there. So I can get O negative blood. O negative blood has no antigens on it whatsoever except for the regular ones. It has no A's, it has no B's, it has no RH factor. So I can get them, and I can get blood from other B's. But I can't get from anyone else. Why can't I get it from AB negative? Right, because it has type A antigen in there. My body would go into attack mode. A couple things I want you to notice here. O negative is the universal donor. They have no antigens on the outside of their blood, as far as we're concerned here, whatsoever. Therefore, there's nothing for anybody else's immune system to recognize, so their blood can go into anyone else. AB positive can get blood from anyone else. If you have AB positive blood, you have type A antigens, so you don't have any anti-A antibodies. You have type B antigen, so you don't have any type B anti-B antigens, and you have RH factor, so you're not going to have any anti-RH antigens. Therefore, you can get blood from anyone else, and you are a universal recipient. Universality does not imply commonality, though. The most common blood type in the United States is O negative. And the least common is AB negative. In fact, all of the negatives across the board are less common than each of their positive counterparts. B positive is more common than B negative. O positive is more common than O negative. Same thing with the ABs as well. AB positive can receive blood from anybody. O negative can donate to anybody. But just as a little PSA here, moment here, Blood donors of all blood types are always needed. It's always way, 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 way better to get a transfusion of your own blood type in an emergency than it is to just get O negative. So please, if you are at all able, I encourage you to donate blood. It is fairly quick. It is almost painless, and it's a really good thing to do for society. You can click this link here to determine if you are eligible to donate blood. Here's a list of the percentages of blood types in the United States. So again, you can see that O positive is by far the most common blood type of all ethnicities across the board in the United States. And look at the discrepancy between positive and negative. O positive and O negative. The negatives are way less, 37% versus 8% in Caucasians. A positive, 33% versus 7% in Caucasians, just to go with the first column because it's easy. On down the line, the positive is always more common than the negative. O's are the most common, followed by A, then B, then AB. Overwhelmingly, though, Americans are O's and A's. I point out that there is some differences in percentages of each population with these ethnicities as far as blood type is concerned. These aren't hard and fast rules. These are super, super, super broad trends. It's somewhat more likely that if you have miscellaneous O positive blood at the scene, that person is Hispanic than Caucasian, for example. If you find A positive blood at the scene, it's slightly more likely that person is Caucasian rather than Hispanic. But remember, that's no hard and fast rule. It's not like there's Caucasians, it's not like Caucasians don't have O positive blood. It's just Hispanics are a little more likely to have O positive blood than Caucasians. As time goes on and our melting pot advances, we kind of intermingle and intermarry more and more and have more children with people from other ethnicities. These trends change and in fact dilute quite a bit. So by the time you're out and working, these numbers might not even matter anymore. The antigen-antibody reaction can be exploited to determine what mysterious blood type would be at the scene. So what you can basically do is you're going to put down mysterious blood sample into a couple of wells. You'll have one that's a control. You'll put blood down in it. You will put a droplet of that mystery blood in a well that's labeled type A, a well that's labeled type B, and a well that's labeled RH. I'm going to put a drop of blood into each. The control you always leave alone. In the A well, you apply anti-sera, which is manufactured antibodies, basically in a liquid, that is designed to attack type A blood. You do the same thing for B with anti-sera that's designed to attack type B blood. And in the final well, you use anti-sera that's designed to attack Rh factor. And what you look for is what's called agglutination. It's going to look spotty like these guys. Okay? Where you have agglutination, that is the blood type. If there's no agglutination, then the antibody was not able to recognize the antigen in the sample, and they're not a match. That's not the blood type. So looking at this example right here, what's that person's blood type? If you said A positive, you're right, A plus. You had agglutination in the A, so the person has type A blood, and you had agglutination in the RH. You have Rh factor. Type A positive blood.